We're going to move on to our next speaker. Deirdre and Gabe will be around for more questions. So next up, we have Cristina Ocana with Washington State University, and she is an agricultural research technician.
Hi, everyone. Yeah, my name is Cristina Gallegos. I'm representing the Berry and Potato Pathology Lab with the Northwest Research and Extension Center down in Mount Vernon, Washington. Today, I'll be talking about the impacts of reduced disturbance and organic matter additions on potato soil-borne pathogens. So Gabe and Deirdre already explained to us what the ELTER project is about. In case you didn't catch it, ELTER stands for Long-Term Agroecological Research and Extension. And as I mentioned as well, our site in Mount Vernon looks at the potato-based annual cropping systems. The general objective of this project is to measure the impact of conservation practices on soil health and farm profitability. Another more specific aim is to look at disease incidence, and we're trying to answer the question, are pathogens present, and if they are in populations that affect crops quality. In this instance, the crop refers to potatoes. Once again, Gabe and Deirdre already explained to us what the treatments are about. But what I want to mention here is that many plant diseases affect potatoes, and to help minimize the loss to diseases, farmers usually use a 4-year crop rotation, and they use the 3 seasons in between to try and to reduce the amount of diseases infecting potatoes. So these 4 different treatments try to mimic what growers do in their fields. And as it was already explained in the previous presentation, all these 4 treatments have different levels of soil disturbance, crop residues, annual cover cropping, and soil amendments. Now, I'll briefly run you through our sample processing. For the ELTER project, we soil sampled the potato plots. We did this by randomly sampling soil from 10 spots at a depth of 0 to 15 centimeters. Then we bulked these samples into a bucket. We took these soils back into the station. We let them dry for a few days, and then we measure 250 milligrams of soil for DNA extraction purposes. This allows us to later quantify the amount of pathogen present in the sample. Then we moved on to qPCR, which stands for quantitative polymerase chain reaction. It's a very long name, but it basically allows us to detect if a pathogen of interest is present in the sample. And with those numbers, we run data analysis to determine if these effects are significant. So at our lab, we work with a few soil-borne potato diseases. One of them is black dot. This is caused by Colihotrichum coccodus. This is a common disease that affects the skin quality and marketability of potatoes. It can be found on stolons, roots, and tubers, and leads to plants being wilted. Once black dot enters a field, it can survive for up to 7 years, and it also can continue to grow in storage with warmer temperatures. We also look at silver scurf caused by Helminthosporium solani. Silver scurf produces blemishes on the tuber surface, which lower the value of the crop in the fresh market. Lastly, we also work with verticillium wilt caused by Verticillium dahliae. This pathogen causes the following symptoms. It causes unilateral stem wilting, as well as browning of the stems and tubers. These damages lead to early crop senescence, as well as reduction in tuber size and hence yield losses. Once verticillium enters a field, it can stay there for up to 10 years, so it's kind of hard to manage. And there are no potato varieties that are resistant to it, but this can be prevented with careful crop rotations. Some of the most common mechanisms for managing soil diseases in potatoes are disease prevention, suppression, and control. Prevention refers to the flora of the land, and so arid or virgin lands are more susceptible to diseases because there's no microbial competition. If a disease enters a field, then we move on to suppression and control. So with suppression, this can be naturally occurring or influenced by management practices. It refers to microbe-based protection that the plants have, and therefore the pathogen does not persist. Lastly, we have disease control. which can be achieved through the addition of organic amendments and crop rotations. So the alter treatments incorporate all of these factors at different levels, and I'm not going to talk much about soil disturbance because we know about the benefits of minimal soil disturbance, but we also know that's not an option for potato. So, but what about the expected effects of the other elements? So for crop residues, these provide a continuous source of feed for soil microorganisms and increase abundance and diversity of microbial populations. For cover cropping, there's the potential for producing compounds that suppress pests and that can act as biofumigants. Both of these are disease suppressive capabilities. As for soil amendments, it can help to inoculate the soil with microbial diversity and suppress pathogenic populations. This is a type of disease prevention strategy. So what does the literature tell us about the effect of increasing soil organic matter on these pathogens? And in here, I'm only going to talk about verticillium because, as you'll see, this is the only pathogen that we were able to detect in our soil samples. But the literature tells us that verticillium readily colonizes sterilized soil, but has very limited activity in natural soil. Also, that adding animal manure contains volatile fatty acids that are responsible for the suppression of certain diseases, including our pathogen of interest, which is Verticillium daliae. Fish emulsions also have a similar effect and can also help to combat common scab of potato. Another one of the elements of the alter treatments is the crop residues. And so one study showed that incorporating millet, for example, into soil had little effect on the reduction of verticillium wilt, though it helped with yields. However, incorporating broccoli helped to reduce verticillium daliae populations, and this is thanks to the release of compounds called glucosinolates. So with all this knowledge, all this background, we know that soil-borne diseases are best managed with increased soil microbial diversity, which is accomplished by cover cropping and organic soil amendments. Therefore, our hypothesis for this study was that the lowest disease incidence would be expected under the alter treatments that incorporate these factors, which are treatments 4, 3, and 2 in that order. So our preliminary results for soil data, for the past 3 years, we did not detect pathogens causing silver scurf or black dot. However, we did detect verticillium wilt. We want to know if the treatments had any effect on these numbers. However, when we look at the variation within each year and within each treatment, we cannot really say that this can be attributed to the treatments. Moving forward, though, we might see effects become significant. As it was already mentioned, next year it'll be the next full rotation. So by next year, it'll be the second time that potatoes go back to a plot. Besides soil sampling, we also quantified internal and external defects of potato tubers. We observed symptoms for black dot, black scurf, and common scab of potatoes. So in 2022, black scurf incidence was very low, ranging from 2 to 9%, whereas common scab ranged from 44 to 57%. In 2023, black dot incidence was 11 to 25%, and common scab was very low, 2 to 6%. Again, there were no differences. These differences cannot be attributed to the treatments as of now. So yeah, in conclusion, the alter will keep looking at the legacy effects of each treatment on disease incidence for soils in the long run. And once again, acknowledging that it takes time to change the biological and chemical properties of soil. And you already know the funding agencies, and yeah, thank you very much. Please let me know if you have any questions.
We have time for maybe one question, 1 or 2.